this morning and have the pleasure of introducing Dr. Cheol for the seminar. Dr. Cheol is an associate professor in the Department of Biomedical Engineering here at Georgia Tech and has a joint appointment in the pediatrics department at Emory University. He recently moved to Atlanta from the Cedars-Sinai Medical Center in LA, where he was an assistant professor at both the Cedars-Sinai and UCLA. Dr. Cheol obtained his PhD from the University of Toronto and went on to do his postdoctoral fellowship at Johns Hopkins Medical Institute in the laboratory of Dr. Eduardo Marban. Throughout his career, he has been investigating how the human heart keeps its rhythm, and his team has been developing biological therapies for heart rhythm-associated diseases. Please help me welcome Dr. Cheol.
Thank you. Thank you very much, Sandra. She's the poster child of our lab, having the faith and vision to join my lab when my lab was nothing more than a little bit of a space with a lot of boxes. So I'm very thankful of her. She's amazing. So this morning I want to talk about how to pace the heart with genes and cells. Of course, we're born with the innate mechanism to pace the heart with genes and cells. And that innate mechanism is shown in this cartoon that actually my cousin drew in about 2 minutes. And each and every heartbeat starts from the sinoatrial node that's colored in red in here. And that anatomic location is at the backside of the heart at the junction between the superior and inferior vena cava that's not shown in this cartoon. And that sinoatrial node, and I'm going to call this one SA node throughout this talk. The SA node harbors specific and very specialized cells called pacemaker cells. And these pacemaker cells do not need any encouragement. They pace, they fire an electrical stimulus every time to keep our heart rhythm. And that electrical stimulus spreads around the atrial myocardium, the right and left myocardium, atrial myocardium, because we're looking at the heart from the front. And then arrives at the atrioventricular node. That's another nodal structure, small nodal structure that sits right between the atria and the ventricles. And with a little bit of delay that happens not by chance but by design, the electrical signal spreads down these fiber structures called bundles. That's the right bundle, and then that's the left bundle of His. And then infiltrates the entire muscular ventricular myocardium through Purkinje fibers. And the anatomical identification of these structures were actually backward. Dr. Purkinje, back in 1845, identified those very extensive networks in the ventricular myocardium. Dr. His identified his bundles, major bundles of the electrical conduction system. And then Tawa, when he was in Germany, identified the AV node. And about a year later, Keith and Flagg in Great Britain identified the sinoatrial node. These are anatomical identification of the cardiac conduction system. The SA node as the pacemaker of the heart was published 3 years later after the anatomical identification. The reason why I put up this historical slide is because the identification of the SA node was done a little more than a century ago. But surprisingly, we know very little about the SA node, and probably because the SA node is so small and a little difficult to access. And this cardiac conduction system going from the SA node to AV node, measure his bundles and the Purkinje system, is universal for all mammalian heart and even in fish heart. So this is a zebrafish 6 days after fertilization. And you can even see, and by the way, fish has not 2 atria and 2 ventricles, but single atrium and 1 ventricle. And you can see an atrium and the ventricle beating together. And the conduction, because they have a single atria and a single ventricle, they don't have His bundles and Purkinje systems, but they do have a sinoatrial node ring at the top of the atrium, and the AV atrioventricular node ring in between the atrium and the ventricle. So that cardiac conduction system is a general a theme in keeping the heart beating in mammalians and amphibians. Now, in also frogs, yeah, absolutely. And in clinical setting, when our heart doesn't beat the way it should, and resulting in abnormally fast tachy or abnormally slow bradycardia, either too fast or too slow, it does the same thing at the end. It sets up an insufficient time for the mechanical events of ventricular emptying and filling. And when the scenario goes worse and worse, cardiac output drops, the lungs become congested, and the circulation collapses. Cardiac arrhythmias are, in fact, the disease that claims our lives at the end. We could have a congestive heart failure, we could have myocardial infarction, ischemic attacks, but at the end, what claims us is the electrical disturbances of the heart, arrhythmias, and it afflicts more than 3 million Americans and account for almost 500,000 deaths annually. And it's expensive to address. $3.1 billion paid to Medicare beneficiaries for all cardiac arrhythmia-related diseases in the US alone. And that was actually about a 10 years old figure, so it's certain to be a little higher now. What are the modern therapies for cardiac rhythm-associated diseases? And there are 3 different ways to approach that. And drug therapy is the first line of defense, and it's variably effective. And the biggest problem with drug therapy is that it addresses one symptom, and it encourages or creates another problem that was not there before. So it's bedeviled by pro-arrhythmic events. And for cases where there's ectopic beats in the ventricular myocardium that shouldn't be, then you can go in and burn it using radiofrequency catheter ablation. And it's potentially curative in ventricular arrhythmias, but less reliable for more complex and most common arrhythmias, such as atrial fibrillation. And these are abnormal activities in the atria of the heart. When all things fail and you need to pace the heart some way, then you get implantable devices. And these things are largely palliative for bradycardias. It could be life-saving for tachyarrhythmias. In all the devices, the patients are committed to the lifelong implantation of the devices and repeated procedures to change batteries once in a while. And it's expensive. And when these things do not work well, you can imagine that catastrophic events can follow. Some of the less common problems are infections. Again, the pacemaker devices, electronic devices are made up of a generator that stimulates or creates electrical shock. And then that electrical signal is related through the wire and has a electrical electrode that gets screwed, literally screwed into the heart muscle inside the heart at the apex of the right ventricular myocardium. So this is the general device implantation strategy. Infections can happen. And for the cases of pediatric patients, it becomes a little more emotional. There are babies who are born with congenital heart defects. And most of the time, these are structural disease. Surgeons go in and try to close up big holes in the ventricular septum. In so doing, they often disrupt the cardiac conduction system, and the baby's heart does not beat very well. So then they implant the pacemaker, and the babies are dependent on, for their life, dependent on the cardiac pacemaker devices. Babies grow, the wires do not stretch. So in the pediatric patients, by the time, and that's the fortunate case, by the time the patients reach up to low teens, mid-teens, they undergo about 4 to 5 replacement of the pacemaker leads. And when I say replacement, they don't take out the old lead. The old leads are still there in the baby's blood vessels. They insert new wires, and the old wires are unscrewed. Because taking out the old wires, it's a little too risky because the old wires are already tissue adhesioned to the vessel. So taking out the old wires, you run into the risk of perforation or tearing the vessel. So in one case that I saw, it was about a 14-year-old boy where the boy had about 4 wires dangling in his heart, old wires, and then only the longest and new wire was pacing his heart. So in those cases, and the adult cardiology, those things motivated us to come up with a device-free way to pace the heart with genes and cells. And what are the potential advantages of biological therapy? Well, pacing the heart with exogenous genes and cells is highly localizable because genes and cells, by definition, the nature is nano and microparticles. They're small. And for pacing, you don't need to deliver these biologicals to multiple areas of the heart. In fact, you don't want to. You want to deliver it in a focal area. So the delivery can be highly localizable. And the therapeutic action is responsible, from the first principle, can be responsible to endogenous signal transduction. And that speaks to something called chronotropic competence. In other words, when we want to run and try to catch a bus, our heart rate has to go up. And that needs to, the biological pacemakers should be and could be responsive to the beta adrenergic stimulation and acetylcholine challenge to slow down the heart rate. And if it works ideally, then it avoids all implantable hardware, and it could be reversed by conventional therapy. And this part is actually an important part because at the very first step of testing this concept in large animals and first in man clinical trials, if it doesn't work as a safety net, we want to reverse it. We want to neutralize it. That can be done quite easily by going back with a radiofrequency catheter and burn the injection site. And I'm going to talk about 3 different strategies in creating biological pacemakers. We call it BioPacer. And these are an ion channel-based approach, stem cell derived approach, and then reprogramming approach. The first one, the ion channel approach, is something that I'm going to go very briefly. And the main idea is that with and I mentioned in another cartoon that the cardiac conduction system begins from the sinoatrial node to pace the atrial myocardium and ventricular myocardium. All these different cells, including the muscle cells, have their typical action potential profiles. These are membrane potential changes inside the cell. The main idea with ion channel approach is to convert a ventricular myocyte action potential to something that looks like the action potential from a sinoatrial node. And there are 2 key differences between those 2 action potential profiles. And those differences, and this is a better cartoon of those, this is a ventricular myocyte action potential, than that this is pacemaker cell action potential from the SA node. One feature of the hallmark of the myocyte muscle cell action potential is that they're really quiet. They don't fire an action potential unless they are stimulated to do so. And that quiet and strongly hyperpolarizing force is provided by an ionic current called IK1. And that's called the inward rectified potassium channel current. So this is getting a little specific for this audience, perhaps. Now, research done by several groups, including ours, about a decade ago, postulated that if one were to destabilize this electrical brake, the cardiac myocytes have everything to pace on its own. And to do so, we created a dominant negative ion channel mutant, replacing glycine, tyrosine glycine, GYG, to triple alanines. And expressing this triple mutant ion channel was able to suppress the IK1 current by 80%. And that was enough to convert this action potential to something that paces on its own. And that was published in Nature back in 2002. Now, that's one way to destabilizing this electrical brake and having myocyte to pace on its own. That's one way. A complementary approach is to overexpress something that's expressed in the sinoatrial pacemaker cells, but not in muscle cells, regular cardiac muscle cells. And that's called HCN channel. And that HCN channel provides a positive current that slowly depolarizes the membrane potential. And that's responsible for this slow depolarization called phase 4 in EP electrophysiology terminology. And that complementary approach was played out for about 5 to 7 years to show that it is remarkably simple to convert the action potential profile from a muscle cell to pacemaker cell. Perhaps what I can show you here is not all the data that we have and other people have published on that theme, but to tell you that we know so much about the ion channel physiology that we could even convert a non-excitable cell like HEK cells to pacing cell. So this was, or fibroblasts even. So we imagined, okay, we'll take human embryonic kidney cells, 293 cell line, and we're going to add some ion channels, a minimal complementary of ion channels that will make the HEK cells to fire an action potential. To do so, what we have done is to add a sodium channel, which is responsible for the fast upstroke of the action potential profile. And then the HCN channel that's responsible for slow rise of the depolarizing membrane potential. And an inward rectified potassium channel. And we had to have that because sodium channel, once it fires, it stays in the inactivated state. So the membrane potential needs to go down for the sodium channel to recover. And to our excitement, having only those 3 channels in the HEK cells made some of these cells fire action potentials spontaneously. So this is a demonstration not how it should be done, but how it can be done. In other words, we're not going to use HEK cells and inject into the patient's heart and use that as biological pacemakers because HEK cells will divide all the time and cause arrhythmia. But it shows how simplistic it is to cause a cell to fire action potential spontaneously. And with all 3 approaches that I'm going to tell you, I'm going to also tell you what are the advantages and disadvantages of each approach. And the advantage of the ion channel approach is that once the ion channel is delivered, you don't have to wait anymore. So you can achieve immediate function. And also, we know so much about the ion channel physiology that the pacing rate can be fine-tuned by making some mutations in the permeation pathway of the ion channel. So a rate adaptation can be possible by modifying the ion channel biophysical properties. Now, a problem with this approach is that I mentioned that if a biological pacemaker works well, then it should be able to speed up or slow down its pacing rate depending on the physiological need. And there's no inherent design element with the ion channel approach for us to do so. And to be able to pace the heart for a long term, one would need to use a genome integrating viral vectors for long-term pacing, such as adeno-associated vectors or lentiviral vectors. I think I spilled my coffee, so I'm going to move. I'm going to clean up this mess after the talk, but make sure that I don't destroy anything here. Okay. The second the second approach to creating biological pacemaker is using pluripotent stem cells. And this is done, thank you, by using embryonic stem cells or iPS cells. And as we know, embryonic stem cells are derived from the inner cell mass of a blastocyst. And by definition, ES cells can be cultured indefinitely and be derived to give rise to all parts of our body. Now, over the last 10 years, the stem cell, the pluripotent stem cell field have moved on and became a mature field to the point that the efficiency of getting cardiac myocytes from the embryonic stem cells or iPS cells can reach almost about 95 to 98%. And this is one of the videos that we collect on a daily basis. Cells that we derive on a daily basis in the lab, where we can make carpets of human cardiac myocytes from human embryonic stem cells. And again, these cells are not sorted, and their intrinsic efficiency to becoming cardiac myocytes is above 90%. And we hypothesize that perhaps we can take advantage of this cardiac differentiation and spontaneous beating phenomenon of these cells. Thank you. And one can make three-dimensional cell aggregates. You can call it cardiospheres or spheroids. And these spheroids beat spontaneously. And one of the reasons why they beat spontaneously is not because they are really pacemaker cells, but they are immature cells. And we reasoned that we could use these three-dimensional spheroids as biological pacemakers once we inject it into the animal's heart. And to do so, we first did an in vitro assay. So we took those spheroids that were beating spontaneously and dropped them onto a monolayer of ordinary cardiac myocytes that are quiescent. And you cannot see it, but you have to believe it. This gray area have that monolayer of cardiac myocytes isolated from a neonatal rat heart. And we did that experiment on a platform called multi-electrode array that can record electrical signals from excitable cells. And this multi-electrode array has electrode endings throughout this platform. And using that platform, you can measure where is the earliest time of activation of the electrical signal. And here you can see the dark color means 0 millisecond, and the lighter color means later, longer time frame. So electrical activation starts from here, where the spontaneously beating spheroids was dropped and spreads to the neighboring myocyte. And when we add some lidocaine, which is a sodium channel blocker, and something that you get when you go to the dentist's office, addition of lidocaine blocks sodium channels and slows down the propagation of action potentials. So this is before and after adding lidocaine. After we add the lidocaine, action potential propagation slows down. The isochronal map becomes more compact, indicating that these two different cell types are electrically coupled. Well, I mentioned that we can get 90% of cardiac myocytes from stem cells, but these stem cell-derived cardiomyocytes are, in fact, a highly heterogeneous population of many different cardiac cell types, including atrial, ventricular, and pacemaker cells. And most of the time, the ratio of these different cell types are in such that atrial and ventricular cardiomyocytes are the major population, and pacemaker cells are the minor population when we derive cardiomyocytes from the stem cells. What we want to do is to convince and coerce the stem cells to give rise to more pacemaker cells and less ordinary muscle cells. And we could achieve that utilizing some transcription factors that are important for the development of the native SA node during mammalian heart formation. And that transcription factor, one of the transcription factors that we have used is called SHOX2. And I'll tell you more about these transcription factors in the third arm, the reprogramming approach. When we used SHOX2 during the differentiation stage, these cells became, higher population of cells became pacemaker cells compared to, relative to the atrial and ventricular myocytes. Taking those cells and not purifying them, these are still sort of heterogeneous population of cells. So this would be a control where the majority of the cells are ordinary muscle cells that do not pace on their own, whereas the SHOX2 mediated differentiation gives us pacemaker cells in its majority. We took those cells and injected into rat heart and wanted to see if those cells will be able to pace the rat heart. And this is an ex vivo. So we would inject these spheroids into the apex of the rat heart. Let the animal live for a few days so that these spheroids will integrate and couple with host myocardium, bring the animals back, open the chest, harvest the heart, and then pace it, and do something called optical mapping. And optical mapping is a technique where you use a photosensitive dye to record electrical propagation. So you can see fluorescence change as a function of a voltage change. And in these hearts, this is where we injected the spheroids. This is control heart. This is SHOX2 cell, SHOX2 mediated differentiated spheroids. And here's the rationale. If the heart is paced by its own natural conduction system, the pacing will come from somewhere in the middle or the side of the heart, because we added some, we cut off the Sinoatrial Node. So the AV node now works as the secondary pacemaker. So So that brief movie shows that the electrical signal starts from here and propagates to the entire myocardium, not where we injected the control cells. But in contrast, the SHOX2 mediated differentiation of embryonic stem cells and injection of those cells at the apex caused the heart to be paced where we injected the cell. And we could, so that was one optical mapping data, and we followed up with surface EKG to show, in live animals, to show that the heart rate was faster in SHOX2 injected animals. And the advantages and disadvantages of that approach is that if this technology matures, we could envision creating an off-the-shelf product. In other words, we create pacemaker cells from the ES or IPS cells and characterize them, characterize their pacing frequency, their longevity, how they react to pharmacology. All those things can be characterized before we deliver those cells into the animal model or to the patient. So off-the-shelf product can be feasible. The problem is, at least with the embryonic stem cells, there's ethical concerns, there's immunogenic problems because these are not the patient's cells. And the risk of teratoma is still a lingering problem. IPS can bypass both the ethical concerns and the immunogenic problems. But the problem with IPS is that you first have to take the patient's skin biopsy, convert them into IPS, and then derive cardiomyocytes. And all that process will take time, number one. And the IPS generation also creates different cell lines with different characteristics from a single patient. So there could be some time and cost related problems with IPS technology. That's not just for this application, but for most of the therapeutic applications as well. The last approach that I'm going to tell you, and this is the most exciting to us, is the reprogramming approach to create de novo pacemaker cells. And I need to tell you something about the Sinoatrial Node one more time. And this is a fascinating structure. If you think about, well, the whole heart has a little more than 10 billion cells. A little less than 5 billion cells are muscle cells. And 5 to 6 billion cells are non-myocytes. And that's what makes up our mammalian heart. The pacemaker cells in the SA node is less than 10,000. So to put this into perspective, less than few thousand pacemaker cells in the SA node can pace the entire heart. So it's a diminutive structure, but highly specialized. And the pacemaker cells in the center of the SA node has slow conduction velocity. In other words, the spread of electrical conduction in the SA node is actually much slower than what you would see in the atrial myocardium and ventricular myocardium. And that's a very important concept in cardiac electrophysiology, and it's called source-sink mismatch problem. And that source-sink mismatch is a problem where the Sinoatrial node, less than 10,000 cells, is such a small structure. How can it pace the huge electrical sink of the atrial myocardium and ventricular myocardium? And that source-sink mismatch can be overcome by slow conduction velocity, which is sort of protective of the Sinoatrial node from the highly hyperpolarizing electrical influence from the atrial myocardium. And the pacemaker cells in the SA node look very differently from the ordinary cardiomyocytes, and I'll show you some features later on. And the last point is how is that the automaticity from the pacemaker cells are formed. And the spontaneous automaticity is driven by a clock mechanism. And there are two clocks. And this has been sort of postulated about 30 years ago, and then fully proven in the last decade that membrane clock and calcium clock, two clock mechanisms drive the automaticity. And to put it in a simplistic manner, membrane clock means ion channels on the cell membrane orchestrate spontaneous action potential firing. And then there is also spontaneous calcium release events that happens inside the cell, and that's the calcium clock one. And that spontaneous calcium release events are mediated by psychoendoplasmic reticulum, SR, in the cytoplasm. And those two clock systems are sort of redundant and gives redundancy and complement each other to give automaticity in the pacemaker cells. Now, it was back in 2007 and 8, and that was a review paper, where Yamanaka published the first IPS manuscripts, and then Jamie Thompson followed a few months later. And then just a year after that, we saw these papers published by a group in Holland. And it was an opportune time, because IPS was new. And then we became to learn that there are very important and specific transcription factors that really guides the development of the pacemaker cells in the SA node, or the formation of the SA node. The mammalian heart starts as a tube, and then it becomes a chamber. So in the linear tubular heart, TBX18 is expressed in the sinus horn, which later becomes, in the mouse life, just a day after it becomes the sinoatrial node. So we knew from these papers, from developmental biology papers, that there are specific T-box transcription factors that are important for the SA node development, ventricular myocardium development, or the outflow tract development. And in fact, when the same group knocked out both copies of TBX18, the Sinoatrial node was gone by about 80%. So we knew that TBX18 has to be really important, but the function of the TBX18 was not known. So being a simplistic scientist, we imagined what would happen if we add TBX18, re-express that transcription factor in ordinary cardiac myocytes. One thing I didn't tell you is that TBX18 is so important for forming the SA node of the structure, but the expression of TBX18 goes away before we are born. So it has a very brief pulse-like expression during embryonic life. In adult life, there's no TBX18. So that's why we imagined that re-expressing this embryonic factor in somatic ventricular myocyte may be able to create pacemaker cells. It was a sort of bold and very simplistic hypothesis. Fortunate to us, it actually worked. So one of the phenotypes that we saw right away was that expression of TBX18 in regular, ordinary cardiomyocyte really shut down a gap junction protein called connexin 43. And gap junctions are tunnels in between cardiac myocytes. And these gap junctions allow movement of ionic ions as well as small molecules. And they are the gates where the electrical communication happens between cardiomyocytes. Connexin 43 is found in general myocardium, both in the ventricular myocardium and the atrial myocardium, but completely absent in the sinoatrial node. So when we express TBX18, it shut down the expression of connexin 43 in the ventricular cardiomyocytes within just 2 days after we expressed it. And here's gap junction proteins have sort of dot-like expression. So here's a punctate expression which is gone with TBX18. And that translated into slow conduction velocity. So this is multi-electrode array recording. In control, the conduction velocity is about 14 centimeters per second. That's how fast the electrical wavefront moves. When we added TBX18, it slowed down a lot by more than 4 times. So in other words, the decreased conduction velocity or slow conduction velocity that are seen in the native SA node could be mimicked by adding TBX18 in vitro. And then we delivered the TBX18 in an adenoviral vector into the animal heart. So we would take the adenovirus expressing TBX18, directly inject into the rat or guinea pig heart, suture up the animal, let the animal live for a few days, a week to 2 weeks, and then bring the animals back. And then study the effect of the TBX18 expression. TBX18 in that vector had a GFP reporter, so green cells indicate the TBX18 transduced cardiomyocytes. And this is sort of, to an electrophysiologist, a money shot, because so this is a technique called patch clamp, where you take single live cells, use a sharp glass electrode, and then have a very tight seal between the electrode and the cell and record electrical activity from that single cell. What we have done is to bring the animals back after a few days, isolate single cardiomyocytes, and we know which ones have TBX18 because the TBX18 transduced cardiomyocytes are green. And when we patch on those green TBX18 transduced cardiomyocytes, they showed oscillating action potentials that are almost indistinguishable from the gold standard, which is the pacemaker cells from the sinoatrial node. So the sinoatrial node pacemaker cells fire action potentials spontaneously and autonomously. This is expanded scale of the same cell. And you can record these cells for 30 minutes, 1 hour, 2 hours until you break the pipette. And we saw similar phenotype in TBX18 transduced ventricular myocytes that we delivered in vivo. In contrast, the control ventricular myocytes that were delivered with a GFP gene alone without TBX18 fired an action potential only when we injected a stimulatory depolarizing ion current. So once it fires an action potential, it stays quiet. as I mentioned at the beginning, ventricular and atrial cardiomyocytes are boring cells. They do not fire unless they are stimulated to do so. So when we saw these electrophysiological properties of TBX18 transduced ventricular myocytes, you can imagine we were really, really, really excited. And what also got us even more excited is that the efficiency of creating these spontaneously beating cells, cardiomyocytes, with a single transcription factor was really high. This is a video of a ventricular myocyte derived, isolated from a young rat heart. And young rat heart, being neonatal stage, they have a little bit of automaticity. So you can see, and this is, these cells are loaded with calcium dye. So whenever calcium spikes happen, you can see cells flashing. And this, in control case, a few cells flash calcium or exhibit oscillating calcium once in a while. In TBX18 transduced cardiomyocytes, it was almost a firework. So the singular re-expression of TBX18 not only creates oscillating action potentials, but the efficiency of doing it in vitro is really high. And another phenotype that I mentioned that the pacemaker cells have, or different from the general cardiomyocytes, that they look very different. And here is a immunostaining. This is from the native heart. So when we take the heart and section the sinoatrial node, and then look at the SA node, which can be found using a antibody against the HCN4 channel, which is found in the SA node alone. We can demarcate where the SA node is. And the SA node is here, surrounded by the right atrial cardiomyocytes. The white staining is a myofilament, a sarcomeric actinin staining. And this myofilament protein is highly expressed in the atrial myocytes, but sparsely expressed in the sinoatrial node pacemaker cells. And when we did in vitro experiment, adding TBX18 in the ventricular myocytes derived from neonatal rat, the highly organized sarcomeric actinin staining or expression pattern was diminished by TBX18, and even the orientation is in disarray, indicating that the muscle-generating, force-generating properties of pacemaker cells are less. In fact, I should have had a video of native pacemaker cell twitching. Pacemaker cells, their function is not to provide mechanical force to the heart. Their function is to generate electrical stimulation, but they do twitch. But the amount of force that they generate is far less than regular cardiomyocytes from the ventricular and atrial myocardium. And here's another immunostaining that we performed on cells isolated from the heart. So we would inject TBX18, and then a few days later, we'll isolate single cells. In control animals injected with GFP vector alone, you will see sarcomeric actinin organization that are beautifully and regularly spaced. In the pacemaker cells, isolated from the SA node, so these are the native pacemaker cells. There's 1, 2, 3 cells sort of clumped together. And these cells are sort of narrow, smaller, and have squiggly ends. And their alpha-actinin organization is in sort of a random orientation. When we looked at the ventricular myocytes that took up the TBX18, their expression was also less in amount and less in organization. And also, they lost some weight. The muscle cells are very plump and large, and these cells became narrow with squiggly ends and tapering ends. And that is very similar to what you would see in native pacemaker cells. And this, I have to admit, is not something that we looked for. The data came to us, and really surprising. And this is something, the morphological change of the cells by TBX18 is something that we are looking into actively. Now, I mentioned that pacemaker cells should respond to autonomic regulation, and this is an in vitro demonstration where we used MEA, and this is a baseline TBX18 transduced cardiomyocytes. When we add beta-adrenergic stimulation with isoproterenol, beating rate speeds up. And then when we challenge it with acetylcholinergic suppression, the beating rate goes down. Here's in vivo data. So we take the guinea pigs and bring back to the lab, and then we make the animal sleepy, and we hook up surface EKGs on its body and record electrocardiograms. And the guinea pigs and rats, their baseline heart rate is about 280 to 320 beats per minute. It's really fast. There's no way we can observe, we thought, the pacing that's provided by the TBX18 biological pacemaker. So we had to slow down its baseline heart rate using chemical. It's dirty, but it does what we want it to do. So by adding methacholine, cholinergically suppress its sinus rhythm. When we suppress it a lot, the animal experiences something called complete heart block, and that's a dissociation of the sinus rhythm and the AV node. So the atrial myocardium is paced by the sinus node, and the ventricular myocardium is paced by the AV node. So, and you can see that here. This large peak, that's called QRS signal, and that's ventricular beat, boom, boom, boom. And then this small beat here, here, that's called P wave, and that's depolarization of the atrial myocardium. And that P wave in normal heart, in my heart and your heart, P wave precedes the QRS wave all the time because atria beats first and then the ventricular myocardium. But here with AV dissociation, the P wave beats at the beat of its own drummer, dissociated from the QRS. That's control animals injected with GFP. In animals injected with TBX18, it shows, number one, the QRS rate is faster than the control animals. And the QRS morphology is negative, downward reflection compared to this guy. And that's by design. We injected TBX18 at the bottom part of the heart. And usually, cardiac conduction starts from the top and spreads to the bottom. And if the pacing comes from the bottom, then it's a retrograde activation, and the polarity of the ventricular activation should be negative, and that's what we see. The third thing that we can extract is this QRS, which is depolarization of the ventricular myocardium, occurs through the cardiac conduction system, through the bundles and the Purkinje fibers, and it's really fast. So it's pencil sharp. TBX18 mediated pacing of the heart because we injected into ventricular myocardium, and it just utilizes normal cell-cell coupling of the regular cardiomyocytes. It's not very fast. So you can actually see a widening of the QRS here. So analyzing those three things, we demonstrate that injection of the TBX18 can create biological pacing in a small animal model. This is a summary of the data where we created ISON, induced SAN-like pacemaker cells. And they beat spontaneously and autonomously utilize the clock system for the automaticity. And the morphology changes, and it's responsive to autonomic regulation. In the last 7 minutes, I'm going to tell you what we have done to take the bench science to something close to clinical realization. And we modified one of the animal labs on the top floor of our research building to something that you would see in clinical lab, in a cath lab. And this is the real lab where Adrian James and Adrian was helping us to do a pig study. So this is a model where we would get about 50 to 60 kilogram farm pigs. And we first created a model in this paper where this is a model where we would first go in with radio frequency catheter. We would first implant an electronic pacemaker device into the pig. And the pig will be paced by the pacing device with a lead at the right ventricular apex right here, not shown in this cartoon. And then we would go in with a radio frequency catheter and burn the atrioventricular node. Basically, you barbecue it. When you burn this one, the sinus node is fine. It's working fine. But that signal cannot be captured by the AV node. So if in effect, you have an AV node dissociation. So the ventricular myocardium then will not be able to pace. So the pacing then comes from the electronic pacemaker device that we implanted. And we had to do it because once you create the AV block, the pig will die. Most of the pig will die within the first 30 minutes. Then we would go in with an injection catheter and inject our TBX18 right here downstream of where we burned. And the idea is if we create an automaticity in the high His bundle region, then that automaticity will be able to use the autobahn of the cardiac electrical system and capture the animal's heart. And that would be much, we demonstrated in this paper, that was much more efficient than injecting the gene into just a general myocardium. And this is one of the cases where large animal experiment is actually easier to do what we want to do compared to small animal, because with small animal model, there's no way we can do this fine localization. Now, one thing is all this procedure is a minimally invasive. There's no open heart. So the delivery of the gene, ablation of the AV node, everything is done by cut down of the right femoral vein. So this is something that is done in clinical situations as an outpatient procedure. That's the injection catheter. You can record the electrical signal with the electrode, and then the needle comes out. We can control it from the other end and inject our gene. Here's the most important data from the animal study. This is control animal injected with GFP. That's pigs injected with TBX18. And it was a 14-day, 2-week study. And about 2 days after we injected the gene, the TBX18 animals started to have mean heart rate that was much higher than the control pigs, somewhere between 70 beats per minute to 80 beats per minute. The resting heart rate of the pigs is about 90, 92 beats per minute. So the biological pacing that we achieved was not exactly the resting heart rate of the pigs, but close. And we were able to maintain that sort of biological pacing for 2 weeks of the study period. And one word here, we set the electronic pacemaker pacing rate at 50 beats per minute. Most of the time, the pigs are paced by its escape rhythm coming from the His bundle area. So in other words, when we have problems with sinus rhythm, we have a slow junctional escape rhythm coming from the His bundle area, and that was a little faster than the device rate that we set up. So basically, the device was a backup pacing.
So I'm guessing that lower right panel, you got the device set up in one of the conditional pacing modes, so it would only pace if a beat was missed?
Precisely.
That's showing in the lower right there?
Precisely, exactly. We wanted to ask the pigs how they were feeling, and we thought one way to ask them is to measure their activity. And the cage was about this big, about 7 by 9 feet. And the telemetry device that we implanted to measure their EKG also had accelerometer that's part of your smartphone. So accelerometer measures basically a vector change over a given time, and that arbitrary mean activity was higher in TBX18 compared to control animals. And when they moved around in bursts, that burst of activity was longer, and they took shorter breaks between those bursts of activity. This is the last data slide, where we wanted to find out where the pacing was coming from. And I mentioned that we implanted cardiac electronic pacing devices and paced the heart from the RV apex. On day one, the day when we injected the gene, both TBX18 animals and GFP animals were being paced from the RV, right ventricular apex, where we inserted the pacing lead, because the TBX18 gene, it takes some time for the gene to express. At the last day of the study period, day 14, the TBX18 pig's pacing was coming from the side of the heart, where we presumably injected our gene, whereas the control animals were still being paced by the RV apex. So in this paper, we demonstrate for the first time that the biological pacemaker concept works in a large animal model. And it's important to emphasize that this is a minimally invasive procedure and right-sided vein delivery of the transgene and injection into the His bundle. The physiological rate, close to physiological rate, was achieved for 14 days. We followed 2 animals up to 3 months, and those animals still had about 70 to 75 beats per minute rate. The main reason why we did 2-week study period was because it was phenomenally expensive to do this kind of study. Cost about $1 million for 30 animal studies. So, and it reduces dependence on the electronic pacemaker device. We think that the first in human clinical trial does not have to be a home run. In other words, we think even if we think the patients who already have electronic pacemaker devices and permanently dependent on it could also receive biological pacemaker genes, and that if that works, it could reduce the dependence of the patients on the device and allow the patients to buy more time between battery change. So that could be first clinical trial that we can imagine. The problems, well, the things that we have to solve with the reprogramming is that the efficiency of reprogramming could be random. We're injecting the gene into the rat, guinea pigs, and pigs, and taking up the gene by the cardiomyocytes is something that are left to chance. The fate of the reprogrammed cells need to be characterized, and this is a problem because there's not a single necessary and sufficient marker for cardiac pacemaker cells. So it's a problem that we think about it on a daily basis, how to solve the problem using and do some lineage tracing experiment. Here's the future, now and future. This is a Sinoatrial Node dissected out from a rat, and we did that maybe 2 months ago, 3 months ago. Loaded with a calcium dye. It's a beautiful structure. It's end to end. It's about 1 millimeter, maybe 2 millimeters big, long. What we have done, we and others have done, is to create biological pacemaker cells in the last decade. We want to evolve away from creating pacemaker cells towards pacemaker nodes, because our heart is not being paced by the cells in a random manner. The cells make up a nodal structure, and it's the node that paces the atrial myocardium. So we want to get there, and what Sandra is doing is to come up with a way to do this in a 2D. And she's working on a microfluidics design where the whole blue and red area will be plated with regular ordinary cardiomyocytes. And then she would flow in TBX18, and by regulating the flow rate, we can make this red area with TBX18 smaller or larger. And we want to see how many cells are needed to be able to pace the surrounding ordinary cardiomyocytes. So that would be a 2D model. And there are 3D models that we are thinking in terms of spheroids as well. So the SA node is very, very small but elaborate, highly specialized. There's a ion channel, stem cell, and reprogramming approaches to creating engineered biopacers. And we continue to try our concept in a large animal model for clinical translation. And that's the future, creating an engineered SA node. And this is the most important slide. These are my colleagues and friends at Cedars-Sinai in Los Angeles, where these people were responsible for the in vitro and small animal model experiments and the work that we published. And then Eugenio Cingolani is my friend who spearheaded and did a lot of large animal studies. And the torch is being carried by Sandra and other scientists in our lab. And here are our collaborators, Dr. Shin, who's in the audience. We are collaborating with her to look at zebrafish model and how that zebrafish model can help us understand the formation of the SA node. And in fact, the zebrafish video that I showed you during the first 2 minutes is the one from Dr. Shin's lab. And with Dr. Charles Zhang, who's also in the audience, we're looking at sort of metabolic differences between the cardiac pacemaker cells and regular ordinary myocytes. And here, conceptually, we think the pacemaker cells should have a little bit of difference in terms of its metabolism. It would, in other words, when we experience hypoxia with ischemic injury, the pacemakers, we cannot afford to lose the pacing ability. So over the course of a few thousands of years, we may have evolved some metabolic differences in the pacemaker cells that are different from the muscle cells. And that work is being pursued with the help of Dr. Zhang. Thank you very much for your attention.
Now we would like to open it up for questions.
Hi, thank you for your talk.
Thank you.
I was curious about the biophysical properties or differences that might exist with these pacemaker cells. You mentioned that they generate less force than the cardiomyocyte. Have you observed whether there might be a difference in their pool is less in terms of amplitude? Or alternatively, it could be because they're a softer type cell, so they don't generate, even with the same pool, they don't generate as much force.
It's that short answer, I don't know, because no one has studied that. But we know from the degree of myofilaments, actinins, myosin heavy chains, alpha and beta, their myofilament expression pattern resembles fetal type, not adult type. So they are perhaps equipped to be less contractile. Whether they are actually softer cells, that's also a very interesting question. They share what begets pacemaker cells. And when we go back, they have some resemblance to so-called second heart field cells, progenitor cells during cardiac development. And the second heart field cells give rise to chamber myocytes as well. So I don't know how soft those cells will be. One hypothesis that we're working on is to see whether pacemaker cells are actually cells that are trapped in adolescence or forever young cells. And that's because when the linear heart tube forms, they begin to beat. And cardiomyocytes, ordinary ventricular atrial cardiomyocytes, have the ability to pace on its own during the embryonic life and up to about late fetal life. By the time we start breathing oxygen, we lose it. So we think we're working on this hypothesis where the pacemaker cells may have some properties that hinder them from maturing further towards working myocytes. Right.
So an important part of a normal, healthy, optimally functioning heart is there are some well-defined, I'll call them spatial or geometrical aspects of the anatomy. So for example, using your pig example, there are many cases in humans when you have ectopic pacing sites or junctional AV rhythms. Sure, the heart's beating and the person's alive, but there's long-term health effects of that heart not operating efficiently, whether it's on circulation, long-term effects of that poor circulation. And to use another example at the beginning of your talk, one of the solutions that exist in mammals for that for that SA to atrial loading problem, and you're probably well aware of this, is that there are fingers of atrial tissue that actually interdigitate the SA node, and that drastic change in surface area is what allows the SA node to conduct to the atrial node. And so I'm throwing these two out examples because I suspect that you and your peers, whether you're pursuing stem cells or reprogramming, probably have to think about or worry about long-term how you're going to deal with these sort of spatial anatomical aspects of cellular reprogramming, how to get the cells in a certain spatial arrangement or where you want them to go.
Oh, that's
Do you have any thoughts about this?
Yeah. That's a wonderful point. And that's precisely what we talk about every day with Sandra and other lab members. As you mentioned in your second part of the comment, the SA node is surrounded by the atrial myocardium. And in the core of the SA node, in the core of the SA node, there are mostly pacemaker cells. In the periphery of the SA node that borders the atrial myocardium, there is a lot of infiltration of atrial myocytes that are going in. That structure, I think it's a little difficult to design spontaneously. In other words, what we may want to do is to have a strategy where we start with TBX18 in the center, and then using microfluidics or maybe some PDMS wafer design, give other transcription factors in the periphery. And the reason why I'm saying this is because TBX18 is not the only one that guides or determines the SA node development. And there's SHOX2 that I mentioned, there's TBX3. So at least there are 3 different transcription factors. What we know now is that during the embryonic life, TBX18 and SHOX2 are contemporary. They pulse and go away, and then TBX3 comes in during the late embryonic life, and then stays with us. So perhaps that has something to do with the spatial organization of the SA node. The strands coming in, the atrial cardiomyocyte strands coming into the SA node, we are at our infancy in sort of designing now.
Copy it, just have to mimic the function.
Hmm?
You don't have to copy it, you just have to mimic the effect.
Yeah.
Some other compensating way.
Yeah. To make the matter a little more, even more complicated, is that the SA node is not pacemaker cells and atrial myocytes. It's got pacemaker cells, and there are something called transitional cells that have atrial myocyte looking like morphology, but they pace on their own. So there seems to be some gradient of cell identity even within the SA node. So the structure is, we have the work cut out for us to study that structure, to say the least. Dr. Shin.
You mentioned that TBX18 has very temporary expression, right, during the development. And if I remember correctly, you mentioned that in adults there is no TBX18 expression.
Yeah, that's.
So, but you think this just one gene, and you can kind of convert the ventricular myocyte to the SAN cardiomyocyte? Why do you think that there is this kind of very transient expressed gene nobody can do this complete reprogramming of cardiomyocyte into the SA cardiomyocyte?
The short answer is, I showed all the data that looks like it's a complete reprogramming, but it may not be. In other words, we haven't really tested the neuronal regulation of the autonomic regulation of the reprogrammed pacemaker cells in full extent. We do not know, again, I mentioned this briefly, we do not know how long the TBX18 reprogrammed cells will stay reprogrammed for months, years. Perhaps it needs the encouragement from the TBX3 as well as SHOX2. And that's the experiment that we are doing now. We are making combinatorial strategies where we do TBX18 with SHOX2, TBX18 with TBX3, all three together in different combinations and different time points to mimic what we see during the embryonic development. And this is data from last week where what we are doing also is take a piece of cardiac tissue from the ventricular myocardium, very small meat, and then just add virus and put them onto a petri dish and observe. And when we do it with GFP, control virus, this tissue just stays as cardiac ventricular myocardium meat. When we added TBX18, they start to twitch about a week later. When we added TBX18 with SHOX2, they twitch within 2 days. So we know that when we combine these factors together, we can increase the efficiency much more.
Is there a dose response to TBX18?
Oh, yeah, that's a good point. We haven't tested it to learn a lot, but what we know is if we have too much TBX18, it kills the cells. At the beginning, thinking that to reprogram, if you take a lesson from IPS, from fibroblast to IPS creation, one wants to infect the cells as much as possible with reprogramming factors. And we did that with TBX18, and we realized it was killing the cells at the same dose as the control GFP virus. So GFP virus at MOI, multiplicity of infection of 10, were perfectly happy. With TBX18, it was killing cells. So we had to tone it down by more than 10-fold with TBX18 to see the phenotype and be able to convert them without killing them.
Is there a endogenous regulator that downregulates TBX18? How does it turn off after postnatally?
Oh, yeah, we don't know. We're sort of standing on the outside and letting the developmental biologists to do the homework. There are some other factors that seem to regulate the Sinoatrial Node program, such as PITX, transcription factors, and that may be an inhibitory, that may impart an inhibitory influence on the pacemaker gene program. Well, thank you very much for your attention.